I cannot start my video, Nidhi.
Are you not able to?
Yeah.
Oh, okay. Oh, that's strange. So I got to, let me just look at it. I think it needs to actually get in as a make co-host.
Okay. Yeah, you should have. Yeah, you can make me co-host. That's better in case your power goes off.
I like the positivity.
Can we make the co-host again? Is it possible after logging in, right?
Yeah, you've already made us co-host.
Okay. Okay. The chat is on.
Yeah, that's fine. Yes, that's been made on. Yep.
The audio is too low. Please log in. Yeah, I mean, you need to activate Professor Kim's video as well.
Yes, I'm just actually making co-host as well.
Well, yeah. Well, to all the students, please log in using your laptop to have a better audio. There are some issues when students, when someone logs in using your mobile phone, the audio is very low.
Okay, and Ellen, I've also made you a co-host as well.
Hello. Voice.
Can't hear you, Alan.
Good evening, Professor.
Can you hear me?
Yeah, I can hear you.
We can hear you well, Professor Kim. How are you?
Very well, thank you. How are you, Nidhi?
Very good. Excellent. All doing well.
I think there's a problem in Ellen's audio, I guess. We're not able to hear Ellen's.
Yeah.
Yeah, she's going to log in again, I guess.
Perhaps, yes.
Should I try and share my screen?
Yes, you can test that out. Yes, perfect. That sounds good.
Oh my goodness. There we go.
Brilliant.
Is that working?
Perfect.
Really? You can see it?
Yes, yes, yes. Well, you can't.
Oh. Okay, maybe I'll move it over here. That's bizarre. Okay.
No, that's...
Oh, well, so if I move this here, oh, that's very good. Okay, so I'll close that. What can you see behind the screen? Anything?
At this moment in time, we can see the screen, but not in a... yes, now this is on full screen. That's correct.
Okay, and do you see also the next slide as well or not?
All moving perfectly well.
Great. Okay. All right, and how do I not share screen?
You can just say stop share. Do you see that? As you take your cursor up, you will see a panel coming down.
Oh, I see. Yes, stop
As
share.
you move it up, then the panel falls down, and then you can just stop share. That's perfect.
Yeah, Ellen has joined back again, I guess.
So Nithi, just for clarification then, I'm going to talk for about half an hour, is that right?
That's correct, yes.
Partly on research and partly on the courses.
Yes, I would have thought, yes, more in terms of the topics, yes, of course, and the courses as well that you could certainly mention. And I think a little bit add on, Ellen is coming over to talk on the courses as well. But certainly you could cover that.
Okay.
But I think hers is more on the other component of biotechnology, I think. So probably
Yeah.
what's in the life sciences is probably where you have it in your presentation, I think.
Okay. Great. And if anybody wants to, you know, if I have to go quite fast or whatever, I can share that with you and you can share that with anybody you feel you would like to. Okay.
Yes, sure. Ellen is making a second or a third attempt.
Can you hear me now?
Yes, Ellen, we can hear you now. We can hear you now. I'm just making you a co-host so you can actually, I think, then be able to click the video on.
Fantastic. Sorry about that. No idea why the laptop isn't letting me connect to audio. Has never done that before. So,
Yeah. Well,
how are you?
Very good. All well indeed.
Fantastic.
How are you, Ellen?
And you are in a perfect month in Scotland at this moment where the sun never sets, isn't it?
It does
Yes.
seem like that. Amazing.
Nidhi, have you made Akshay the co-host?
I will make him a co-host as well.
So do we know how many people will be in the audience?
Yeah, we got, it's a very interesting and very positive news. So we got 500 registrations, more than 500 registrations, and we are expecting around 150 to 200 participants.
Wow.
Yeah,
I know.
and we have got participants across India this time, and that's one of the reason is we are, I mean, hosting this along with Crescent Incubation Cell. It's considered one of the top 5 incubation cells in biotechnology in India. So there has been a wider reach, and Mr. Parviz will be joining the CEO of Crescent Incubation soon.
Wow,
Oh
amazing.
my goodness.
So the 2-member team who are here. Akshay is there, who is pretty reluctant to show himself at this moment, but I'm sure he will very shortly. And as well as his head, Parvez, will also be with us. So the two are a part of the incubation center. They've been quite instrumental in getting this information across to every part of India that we are aware of.
Hello everyone, Akshay here.
Yes, hi Akshay, how are you?
I am good. Sorry for not able to turn on my video. I am having a low bandwidth issue, so you'll either hear a robotic voice if I turn on video. So chosen not to.
Ah, really? It's not because you felt that you will be lower in competition in terms of the looks with us or anything like
that. Kadir is the best looking, so obviously I cannot beat him.
That's okay. Well, we'll take your word for it, although I'm a bit doubtful on that one, but that's okay.
So I hope everything is fine. The presentation and everything is working fine.
Yes, we did do a screen share, and I mean, anyway, you're there as a co-host. I think the recording kind of started a little earlier on. Would it be a good idea to just?
You can pause it for the time being.
Okay, that's fine. So I'll just do it at this.
Good evening. Good evening. Good evening. Good evening to all joined from India, and good afternoon for people joined from UK. And it's very privilege and honor to have Professor Kim for a joint webinar with CIC and Scotsgrad. And I thank Scotsgrad for this opportunity, especially Nidhi and Kadir for this. And so I just quickly go through Professor, has a lot of, Professor Kim has a lot of very detailed profile. Maybe I'll just quickly go through. I think she has a very wide international experience, starting from, so she is currently the Associate Dean of International and Research Scientist at the School of Life Sciences at the University of Dundee. Kim is also a guest professor at Taylor's University. She undergraduate and postgraduate degrees she got it from England, and did a postdoc in France and as well as USA before coming to Dundee. So you speak, I hope, some French too. So Kim is internationally renowned leading scientist in the role and regulation of the notch signaling pathway, which plays essential roles during the vertebrate development, which also underlies many diseases. In the talk today, she describes she's going to describe the techniques she uses to manipulate gene expression and gene function to monitor the role of candidate signaling pathway in building the embryonic body plan. So with this introduction, I welcome Professor Kim. Actually, Kim, just to give a quick overview, we are BioNest is the logo which you see, the BIRAC. It is the Ministry of Science and Technology, Department of Biotechnology, entity. We are funded by them. We have 22 life science startups in our incubation center. In fact, some of our startups also would have joined today. Plus we also have a School of Life Sciences and School of Pharmacy. So apart from this joint webinar, we invite you to mentor our startups, as well as we can do a lot of joint collaboration, with our university, and we can participate along with your university. With this introduction, I welcome you again, and then I leave the floor back to you. Thank you.
Thank you very much, Parvez, and thank you for that very kind introduction. And I'm very excited to be participating in this fantastic event today, and I look forward to working closer with you in the future. Thank you very much for that suggestion. So I think without further ado, I will just start my presentation. Could we just have confirmation everybody can see that?
Perfect.
I get it into, yep. So just to start off then, please do share with all of your friends and families and colleagues that you're with us today. And I'll just start with this beautiful photograph, which is a photograph of the Dundee Law Memorial, just sitting there on this hill at the top of the city. And as you can see, the city of Dundee sprawling out beneath that on the edge of the beautiful River Tay. So an absolutely fantastic location that we're talking to you from here today. And as an institution, the University of Dundee has a core purpose that I've outlined here, and that is to transform lives both locally and globally through the creation, the sharing and application of knowledge. And of course, that rings very true within the discipline of life sciences. So as you heard from the very kind introduction, my name's Kim Dale. I wear a number of hats at the University of Dundee. Today I'll be talking to you about the work in my lab as a developmental biologist. But as you heard, I have a number of other roles, which is that I'm the Associate Dean for my school. So the Associate Dean International, in charge of all international partnerships, from which I hope we can develop more today. And I'm also the University Regional Lead for Southeast Asia. So I'll talk to you today about developmental biology and what we've been learning about a particular pathway, the Notch pathway, and what role it plays in this context. So developmental biology describes how we all go from a single fertilized egg to this exquisitely patterned organism on the right, where everything develops at the right time and in the right place. And that really is quite a miracle. I'm actually quite a good developmental biologist because this is one that I made myself, and her name is Monica. So this very complicated process, we break down into a number of steps. So we start as one cell, and that individual cell differentiates to form a whole variety of different cells, so a total of 200 different cell types that are required to build all of the tissues of the embryo. And those different cell types form by communicating with one another. And so I'm interested in understanding how cells talk to one another to build the tissues and organs of the body. The reason that's so important is not only to understand development, but those same communication systems, if they go wrong, give rise to disease. So the building blocks then, a very sort of swift walk through development, of course, we're all made up of about 37 trillion cells. In each of those cells, there's a nucleus. In each of those nuclei, there's 26 pairs of chromosomes that each are made up of DNA that are compartmentalized into little genes. And it's the genes that are on or off in any one particular cell that tells that cell what type of cell it is. So for example, here's a skin cell and a bone cell represented by these squares. Here's the DNA and those little blocks of DNA called genes. And the difference between the skin cell and the bone cell is the genes that are on in green or off in red. And you'll see that combination here in this simplification is that the order of genes that are green or red are different. So how do cells know which genes to turn on and off? Well, they communicate with one another. So one cell sends a signal to another, and that then changes the genes that are on or off. So in this context, here's the signaling cell. It releases a signal here in red that's caught by this little Y-shaped protein on the receiving cell membrane, and that protein is called a receptor. So once the receptor receives that signal, then you change the genes that are on or off in the nucleus. And not all cells respond to all signals. For example, here, that receptor can't receive the signal because it's the wrong shape. So once a cell receives a signal, it can do a number of things. It might, on receiving signal B or C, it might choose to move, it might choose to survive or divide, or perhaps differentiate, which means become a different type of cell, or in fact, on receiving none of those signals, it may die. So I'm interested in one particular communication system, one particular signal, and that's the notch pathway. And depending on the time or the place or when in development a cell receives the notch signal, it could do one of all of those things we talked about. It could proliferate, differentiate, die, etc. So notch signaling mediates cell fate choice, division, or survival in many processes in development. I'm interested in its role in neural development and the development of the skeleton, but there's all these other tissues that notch plays a very key role in the development of all of those tissues. And in fact, when notch goes wrong, it can lead to developmental defects as well as a whole host of different diseases. And here's a whole host of different cancers that have been associated with abnormal notch. And right at the end of the talk, I'll come back to indications that we've got from the role of notch in development that may implicate it in breast cancer. So looking at the notch pathway then, similar to the schematic I showed you earlier, here's a signaling cell and it releases this signal in purple called jagged or delta, and that signal is caught by this receptor in the responding cell, and that receptor protein is called notch. When that signal is caught by the receptor, this little red part we call the intracellular domain, so notch intracellular domain, which we call NICD, that little piece of the receptor is cut off, it goes into the nucleus and turns on genes. So it's very important then to make that NICD, to cleave that receptor so the red piece goes into the nucleus, but it's also really important that we get rid of it efficiently. So it's a careful balance between producing the notch and getting rid of it. We know that's true because there are diseases like this disease, T-cell acute lymphoblastic leukemia, which mainly affects children. 50% of those patients only have that disease because they have too much of that red bit of the protein. They don't degrade it. So if you have too much notch, that can lead to disease. So that means it's important to understand how we get rid of notch, how we degrade it. And we actually know very little about that. What we do know is that when ICD goes into the nucleus, then it recruits a degradation complex that degrades it. But we don't know what controls spatially and temporally when that degradation happens. So I look at that, but I look at it in development, but of course I don't use human embryos for development studies. I use the chicken and the mice, and you might think that's a bit crazy because the body plan looks so different. But actually, at very early stages, the body plan is very, very similar. So here's a human embryo after 4 weeks of development, and here's a chicken embryo after 2 days of development in the egg. And you can see that they're very, very similar. So to understand body plan formation and the role of signaling in developing and making the human body plan, we can use the chicken embryo as a model. And so at these early stages of development, there is the tissue here that will give rise to the spinal cord and the brain. And here are little segments we call somites that even at 2 days of development are apparent. And those segments are what will give rise to the segments of the skeleton. So we can study the body plan formation of the two tissues I'm interested in, the neural tissue and the skeleton, by studying early stages of chick development. The other thing about the chick, which is remarkable, is that things take place very quickly. So that's a 2-day embryo that I just showed you. It looks like very little to look at, actually, but in 24 hours, a huge amount of development happens, and those cells of the embryo do lots of the things we just discussed. So in 24 hours, cells move and differentiate and migrate, some of them die, and so that in a 24-hour period, you go from an embryo that looks like this with very little morphology into this beautiful 2-day embryo. So in order then to look at the role of notch, there's one particular cell type that I'm interested in looking at, and that's a stem cell. So a stem cell is remarkable because it can give rise to any type of cell, it can differentiate to any type of cell, and it also can make more of itself. So I'm interested in looking at stem cells, which give rise to the two tissues of interest, the neural tube and the spinal and the skeleton. So we identified those cells because we can label them with a green fluorescent protein. So here's a 2-day embryo with those little stem cells labeled in green. And if you culture that embryo overnight, those stem cells divide and their daughters either give rise to these somites, which will give rise to the skeleton, or the daughters go to this neural tissue. So those stem cells make a choice, a cell fate choice, to become skeletal tissue or neural tissue. So to know whether notch plays a role in that cell fate choice in those stem cells, we took notch away, we blocked notch signaling in those stem cells. And what we see is that now those stem cells no longer go to these segments that give rise to the skeleton. Those stem cells only go to the neural tissue. So that tells us that notch plays a role in cell fate choice in stem cells. Without notch, you do not build this skeleton. And this is just showing you the wonderful PhD student that published that work, showing that notch makes stem cells choose where their daughters go. I haven't got time to go into lots of data, but basically this is a PhD student that showed other signaling molecules that help those cells remain as stem cells. And these wonderful PhD students helped us understand that notch works with another pathway called sonic hedgehog to allow us to develop different types of neurons. I'm now going to switch to talk about work that we've been doing to look at the role of notch in that other tissue, the tissue that gives rise to the skeleton. So all vertebrate species, although they have an adult form that looks very different, they all share this conserved feature that they all have an internal skeleton. And this is a very important structure because it gives you both rigidity as well as flexibility. And there are many congenital vertebral malformations where the skeleton doesn't form properly, and we know very little about what happens in the formation of those malformations. But there's one family called spondylocostal dysostosis, and this is a family of pathologies where 5 genes have been identified that underlie the disease, and all of those genes belong to the notch pathway. So that suggests that notch signaling, the notch signaling between cells, is important to develop the skeleton. So we did an experiment to test that hypothesis. So here's a beautiful mouse embryo or a chicken embryo with those lovely segments that will give rise to the skeleton. But if you culture a mouse embryo or a chicken embryo with no notch signaling, you see that those segments are completely absent. So Notch is essential. If you don't have notch signaling, you form no segments. So I'll tell you a little bit more about this process. So those segments that I've been describing to you form by chopping off the top end of this unsegmented tissue. That's what's shown here in schematic. So there's an unsegmented tissue that becomes segmented gradually over time to form these somites that will give rise to the bones of the skeleton. And the timing of that process is different in different species, and it's regulated by dynamic gene expression, which is visualized here. So there's the unsegmented tissue that has dynamic gene expression shown in green. And what we think is that that dynamic gene expression acts like a clock in that tissue, telling that tissue when to segment. So we've done a lot of work to show that production and degradation of notch signaling is important to regulate the timing with which you make those segments. And these are the wonderful postdocs and PhD students in the lab that did that work. But what we didn't know is how degradation of notch is regulated. So that comes back to what I told you at the beginning. So it's important to make notch and get rid of notch. And we've shown that's important to regulate the timing of embryogenesis. But how do you regulate when you degrade notch? Well, what I told you is that this notch receptor, here it is cleaved, and that little intracellular piece goes into the nucleus. And what we've been able to show in the lab just in the last year is that a label is added to that protein, which we call a phosphate. So we phosphorylate that protein, and that recruits the degradation complex that degrades NICD. So here's NICD, and we've shown that all of these labels are added, but this red one is important in order to degrade notch. And that label is added by two proteins called CDK1 and CDK2. Once they add that label, NICD is degraded. But the final piece of exciting data I want to share with you is that CDK1 and CDK2, the proteins responsible for degrading notch by adding that label, have another important role in the cells. So cells grow, they duplicate their DNA, and then they prepare for division, and they divide. And as cells go through this cycle, grow, DNA synthesis, and then divide, that cycle is regulated by the activity of CDK1 and CDK2. So as you go round this circle, levels of these two CDKs go up and down and up and down. So when levels are high, you move to the next phase of the cycle. What we've shown is that as you move through those phases of cell division, levels of notch protein also go up and down, because these proteins add a label that degrades notch. So when CDK is high, NICD is low. When CDK is low, NICD is high. And so what we've shown then is that notch turnover is regulated by proteins that regulate cell division. And cell division is essential to making an embryo through development, but it's also important in disease like cancer. And so we are now investigating whether the regulation of notch turnover is playing an important role in breast cancer, because many patients that are treated with breast cancer treatments and that then escape treatment and so have a relapsed cancer also show elevated notch. So we're trying to see if the link between cell cycle and notch is the reason behind patients relapsing from breast cancer. So at that point, I'll just summarize what I've told you, which is that we have shown that this pathway, the notch pathway, tells stem cells in the embryo whether or not they should become spinal cord or skeleton. I haven't had time to show the data, but we also show that when we already have the spinal cord, notch acts in that neural tissue to decide which different types of neurons are made. Notch plays an essential role in building the skeleton, because without notch there is no skeleton. And finally, in very recent years, we have shown how to turn this very important pathway off. And we think that that will play a very important part in our understanding of the role of Notch, both in the development of different tissues as well as in different diseases. So it just remains for me to thank all these wonderful funding societies that have awarded me grant funding, my fantastic collaborators in the School of Life Sciences and the School of Engineering that I collaborate with. And this is my wonderful lab at the moment, which I have to say is all female. And I think that says a wonderful thing about science these days. And I will finish there. I don't know whether or not perhaps the hosts would like to tell me whether I take questions now or whether I go on and discuss a little bit about the courses.
Ms. Kim, I think we are just getting people to ask questions on the Q&A. So I think it's good to just finish at your end in terms of the questions while they can think about certain questions that they may have. So I think it's best to proceed and complete at your end.
Okay, wonderful, thank you. So what I'll switch to now is to talk to you a little bit about the different types of postgraduate courses that we have in the School of Life Sciences for training the next generation of postgraduate research scientists. So the courses that we have are the following. We have some taught postgraduate master courses. So these are courses where you have 3 semesters over a year, 2 semesters are taught modules, and 1 semester is a project. And I'll go into more detail about those in a minute. We then have our research postgraduate courses. One of those is the Masters by Research in Life Sciences. That is also a 1-year course, but rather than having only 1 semester in a lab, you spend your entire year doing a research project. So there are no official taught elements, it's all lab-based. That's the difference between the Masters by Research or the MSCs. Finally, I'll talk to you about our PhD programs. So if we start with the taught postgraduate, I'm just going to start first of all by describing why you might want to think about Dundee as a destination. So we're one of the UK's top 30 universities. We're 4th in the UK for student experience, and we're 4th in the UK for overall average satisfaction in the International Student Barometer. We've got a TEF Gold for our teaching performance, and we are also University of the Year 2020 for student experience. The School of Life Sciences is the top research institution in the entire UK for research in life sciences. So we score more highly than Oxford or Cambridge. We are #1 in the UK, 26 in the world for research impact, and we're the most influential scientific research institution in the entire world for pharmaceuticals. This is just one of the recent discoveries made by one of our 10 research divisions, and that is a new anti-malarial therapy that's currently in clinical trials. For our teaching, we're 3rd in the UK for biological sciences, just behind Oxford and Cambridge, and that was in the very recent Sunday Times University Guide, but also in the Complete University Guide 2021. We're top in the UK for student satisfaction for a number of our courses, and 79% of our graduates are in graduate level employment within 6 months. So on to our taught postgraduate master's programs. We have either biomedical and molecular sciences, or you can do that same course but combine it with business, entrepreneurship, management, or marketing. Why then would you want to think about these types of courses? Well, the answers are that you would be learning about biomedical and molecular sciences in, as I say, one of the world's leading centres for biological sciences. You'd get exposure to industry standard technologies and equipment. And we also offer you these business modules which prepare you for a role in the biotech and pharmaceutical industries. We are innovating in a number of areas in our research divisions. So these are the research divisions in either the School of Medicine or the School of Life Sciences in which you would be able to do your research project. The programme focuses on molecular mechanisms in disease, looking at the genetics, epigenetics and genomics of disease and cell biology, therapeutics, advanced technology platforms and professional skills, as well, of course, as your research project. The structure, as I said, is 3 semesters. You do 3 taught modules in the first semester, 3 taught modules in the second semester, and the third semester is your research project. A typical week has about 2 2-hour sessions, and those are lectures in each of those modules in semester 1 and 2, sprinkled also with some workshops of 3 to 4 hours. So you have between 6 and 12 hours contact time a week, but a total of 200 credit hours. So if you chose to study one of our TPG programs with some of those business-related pathways, this gives you the opportunity to, instead of doing a wet bench project in that third semester, you do a bio-business project, focusing on business or marketing ideas aligned with a bioscience research project. So as I said, you could choose to do that project, you could choose to do that program with business, entrepreneurship, management, or marketing, and the course structure looks very similar. It's just that we swap 2 of those theory modules. Instead of being based on basic research, you would choose one of these modules in that first slot and one of these modules in the second slot, depending on whether you're on the business, the entrepreneurship, the management, or the marketing route. These courses are all very much designed to fill skills gaps that have been identified by future employers of our TPG programs. So we really are endowing students with the knowledge and skills that employers are looking for to make sure that you are prepared for a really meaningful career directed towards solving real-world problems associated with human health. So the courses are designed to equip you with the skills that employers are looking for, having researched and spoken with these employers. The course reflects current research themes in our life sciences school. You're learning from world-class researchers at the cutting edge of their fields, and you're experiencing labs and facilities that really are state-of-the-art. For the research postgraduate options, as I said, you could choose to do a master's by research. Again, this is a 1-year course, but it's entirely based in a lab. So you would choose your top 3 projects from the list of projects listed. You would be interviewed by myself and the PIs of those projects. And if you were successful at interview, you would then join an induction program with the PhD students for the first week or 2, and then carry out a research project for a year. At the end of that, you would have the option to think about upgrading to a PhD program if that's what you wanted to do. So PhD students, and in fact all of our master students, have access to outstanding core facilities such as quantitative proteomics. We have the biggest number of mass spectrometers throughout Europe. We have sophisticated image analysis tools, computational biology. So no matter which route your project takes you in, we've got fantastic facilities to allow you to be sure that you really are at the cutting edge of your field. Sorry. So for our postgraduate research training then, you are exposed, as we've said, to high quality research experience. You have both a primary supervisor and a secondary supervisor and a thesis committee for the Masters by Research and PhD programs. Your writing up of your thesis is central to the award of your degree. And as I said, you could start out on a Masters by Research and then upgrade at the end of the year to one of our PhD programs that are either 3 years or 4 years. You could alternatively apply directly to our PhD programs. So those PhD programs, we are aiming to train and support students for an impactful future, giving you a career in academic research as one possibility, but also giving you a skill set in all of these different types of skills that will open up doorways to opportunities beyond academia. So in your first year of studies, whether that's Masters by Research or PhD, you engage with an induction week that includes courses on ethics, unconscious bias, time management, calculations, statistics, as well as some compulsory training in scientific writing and research integrity. There are also then elective elements that you can choose to do, a whole host of different professional development courses that are open to all of our postgraduate students. We have a fantastic international student support for all of our international students, but I'm sure that Ellen will pick up on that when she takes over from me in just a few moments. And finally, to end then, the output and impact of our postgraduate courses is to really develop creative and confident interdisciplinary graduates with very strong quantitative skills, educated with a deep knowledge of biological systems in animals, plants, microbes, both in the health and the disease context, and projects aimed at trying to develop effective and safe treatment for diseases. With that, I'll finish and take any questions. Thank you.
Brilliant. Thank you very much, Professor Kim, for that presentation. We've got a couple of questions here. I just hope that some of these questions don't take 2 hours to answer. They are just appearing on the Q&A section over there. Is it better that I read it, or is it better that you read it? Which is more comfortable?
Right, okay. Well, I could read one and then maybe we'll alternate. Does that work, Niti?
Yeah, that's fine. I'm okay with any. It's just that life sciences is not my cup of tea, but I might pronounce certain words wrongly.
Maybe by the end of this, we will have changed your view on that. So I can see one question here is asking about the T-cell acute lymphoblastic leukemia. And is there any treatments for the fact that we have, aberrant notch degradation? Well, that's an excellent question. And of course, yes, that is an area of current research. And when I described at the beginning the way that the notch protein is cut so that the intracellular bit goes to the nucleus, there are now drugs that have been developed that stop that enzyme that cuts that intracellular piece so that it reduces the production. So the degradation is still bad, but because we're reducing production, it's a different way to combat that disease. So that is used in some contexts. The problem though with that is that unless you very carefully monitor where you're adding that drug, you will be preventing notch signaling in other tissues as well. So it's this very careful balance of making sure that you only affect the tissues of interest with those drugs. But it is an area of development. You just have to also be careful of off-target effects. Could I provide more insight on the notch pathway in terms of different cancers? So actually, in that list I gave you, most of those cancers come about because there's too much notch, because it's not being degraded properly. And that's why my lab focused on trying to understand better how that's normally supposed to happen. And so what we're hoping is that this new piece of the puzzle that we've just published in my lab, showing that the CDKs regulate addition of that phosphate, which then drives degradation, we can now maybe target where and when those CDKs are acting and maybe push them to act more effectively in areas where NICD degradation is not happening. And that will be looking in the context of those different cancers. Right, okay. Is it canonical or non-canonical? That's an excellent question. These are fantastic quality questions. Thank you very much for those. There is a non-canonical pathway that's very much understudied, and I think that probably does play a very, little understood role both in development and disease. The pathway that I study, though, however, is the canonical pathway.
And the next question, I'll read out for you. I should do my part as well, isn't it? How do you detect notch defective pathway in the human embryo to prevent spinal deformities?
Another excellent question. So the way in which you can see if a pathway is active or not is to look at the expression of those target genes. If you remember, I said that when notch goes into the nucleus, it turns on target genes. So you can monitor the expression of target genes, and if they are not on in the way that they should be, that suggests notch is not acting properly. If they are expressed at too high levels, it suggests that notch has not been turned off.
Right. Does notch is involved in any other cancer? That's the question.
Yes, so
some diseases where Notch plays an important role.
yes, so if I just go back to one of my slides, it's actually quite scary how many different pathways Notch does play an important role in. So it plays a role in pancreatic cancer, cervical cancer, skin cancer, medulloblastoma, ovarian cancer, prostate cancer. And that is known because the target genes or that piece of the Notch receptor is either the levels of expression are too high or too low compared to control patients. So what that suggests is that it's playing a role, but we don't know what role it's playing in many of those diseases, and that's the work that's currently underway.
Right. And the next question. Right. That's a question with regard to chick embryo instead of humans, but after a certain weeks, there might be possibilities that their functionality changes due to differences in signaling system. So at what point you can identify these things? That's the question.
So that's a very good question as well. So what I started out with at the beginning was to say that, you know, obviously we can't use human embryos to try and understand the role of these signaling pathways in developing the body plan. And animal models such as the chick and the mouse are excellent, but up to a certain point. So in actual fact, we only study these processes at very early stages of development where there is high degrees of conservation. At later stages, of course, things go very species specific. So developing, for example, feathers or wings or fur or, you know, all of the different things that are species specific. So the use of animal models is really restricted to very early stages of development. So before the chick has hatched, for example, while it's still in the egg.
Right. Is aging and related disease? In aging and related disease, why the stem cells are not able to function as a normal condition?
Sorry, could you just repeat that one a minute? Sorry.
In aging and related disease, why the stem cells are not able to function as in normal conditions?
An excellent question, not one that my lab particularly follows because that is at the other end of life. So I very much look at the sort of the beginning of life and body plan formation. But what we do know is that Notch does play a role in neurogenesis, so in the formation of new neurons. And so it is extremely likely that in, in those aging processes where you still got stem cells in the developing brain and in the adult brain, the Notch plays a role, but I'm sorry to say that my lab doesn't work on that.
Right. Well, there are some questions which are related to internships, admissions, and research degrees, and so on. We will take them on later on. I think that's something that we could obviously handle later, so that's not a real problem, and I think we'll get them answered later. What procedure is followed in developing tissues in lab?
What procedures? Right. So what I sort of touched on a little bit today is that if you want to try and understand the role of a signaling pathway in its normal context, you need to be able to disrupt gene function only in that tissue, right? Because if Notch plays a role in pancreatic development, breast development, neural development, skeletal development, and you're just interested in the role, for example, in those stem cells, what I showed you was an embryo where we just labeled the green cells in those stem cell region and disrupted Notch in that region. So the way that we do that is a procedure called electroporation. We introduce DNA that is either control DNA or mutant Notch DNA, and we ask what happens to those cells. So electroporation, gene manipulation. We are also now able to get those stem cells. You may have heard of induced pluripotent stem cells. These are stem cells that you can differentiate into any type of tissue in the dish. So now in vitro, no longer needing animal models, what we're able to do is to take a stem cell and make that presemitic mesoderm, the tissue that makes the skeleton, and we can do that in a dish. And what that means is that we're going to be able to avoid animal experimentation and have quantities of cell material that allow us to really look at the biochemistry at the molecular level of what's going on.
Right, and there's also a question on, could you explain a little more on the role of Notch in inflammatory reactions?
So in immunology, Notch plays an incredibly important role. And actually, in the T-cell acute lymphoblastic leukemia scenario, the reason that those patients become so ill is that Notch plays a stem cell fate choice role there in promoting the T-cell fate over the B-cell fate. So particularly in the role of inflammation, I'm afraid, again, that's not my area of research, it's not my forte, but I know that Notch plays a role, sort of, as I'm saying, in sort of cytokine responses and in cell fate choices. So again, a primary role for Notch signaling at various levels and various steps in an inflammatory response.
Right, brilliant. What triggers Notch to be produced in large quantities?
Well, so if you remember, the way that Notch is activated is that it has to catch that ligand from the adjacent cell, and that protein is called jagged or delta. So if there are many cells surrounding that receiving cell that have high levels of delta, then all of the Notch receptors in the receiving cell will be activated, and so then you'll generate lots of that intracellular protein. So it's partly dependent on the number of adjacent cells that express the ligand, and it's partly dependent on the number of Notch receptors that are in the receiving cell.
Right.
Okay. So, Ms. Nidhi, can I just make a quick note? So, Professor Kim, this has been one of the most interactive Q&A sessions. As of now, we stand at 34 more questions. Yeah. Yeah, so in the interest of time and to ensure that all the proposed agenda is covered, I would encourage the participants, if they can mail in their queries. If somebody from the Scots grad team can share their email ID in the chat window, I'll encourage the participants to put their questions over email, and we'll ensure that those questions are answered. So over to Nidhi sir with the further proceedings. Despite the fact that we would like the Q&A to go on, but there are too many questions. So in the interest of time.
Yeah.
Lovely. That's fine. Thank you very much, Akshay. We do understand that. So let's move on with our part of the presentation. And,
I would just like to say thank you very, very much. And I'm absolutely delighted there are so many Notch biologists in the new generation of science.